Hey everyone in Mr. Martin's class. Welcome to the deep dive. Today we're diving into the fascinating world of nucleic acids.
That's right. Get ready to explore the amazing molecules that make life possible. DNA and RNA.
So, no pressure, but we're basically talking about the instruction manuals for building and running every living thing.
You got it. And we're going to break it down in a way that's clear, memorable, and, dare I say, fun.
Perfect. Because let's be honest, sometimes textbooks make this stuff feel a little dry.
Absolutely. So let's ditch the jargon and make this deep dive feel more like a cheat sheet for understanding the really cool stuff that makes life tick.
Okay, let's start with the basics. What exactly are nucleic acids?
Well, imagine a language that tells your cells how to build you from the ground up.
Okay, so DNA and RNA are basically the blueprints and the messengers that make life happen.
Exactly. And like any language, they have their own alphabet or building blocks. We call them nucleotides.
Nucleotides. So are those like the letters in the DNA and RNA alphabet?
Yes. And just like letters have their own unique shapes, each nucleotide has a specific structure. It's made up of a sugar, a phosphate, and a base.
So it's like each nucleotide is like a tiny Lego brick and you can build anything with them?
Great analogy. Now let's focus on DNA.
DNA, the superstar of genetics. The molecule that holds your genes.
Right, exactly. It's the instruction manual that makes you you.
Hold on, I've heard that some viruses actually use RNA as their genetic material.
You're right. Some viruses are kind of the rebels of the biological world. They do things a little differently. But for most organisms, DNA is the main molecule in charge.
Okay, so for most living things, DNA is the boss. What makes it so special?
Its structure. DNA has this incredible double helix shape.
A double helix. What does that even look like?
Imagine two spiral staircases winding around each other, connected by steps. Those steps are the bases, and they pair up in a very specific way.
So it's like a puzzle where the pieces only fit together in a certain way.
Exactly. Adenine A always pairs with thymine T, and guanine G always pairs with cytosine C. We call them complementary base pairs.
Got it. A with T and G with C.
This pairing is crucial for a couple of reasons. First, it makes DNA super stable, like a fortress protecting the genetic code inside.
So the information stays safe.
Right. And second, it allows DNA to replicate or copy itself perfectly.
So when a cell divides, the DNA basically unzips.
Exactly. It unzips down the middle, and each strand acts as a template to build a new matching strand.
So you end up with two identical copies of the original DNA molecule. That's incredible.
That's how genetic information is passed down from one generation to the next with amazing accuracy.
Wow, that's mind-blowing. So DNA has this amazing structure for storing and copying genetic information, but where does RNA fit into all of this?
RNA is like DNA's multi-talented cousin. It's single-stranded, so it's not a double helix. It also uses a slightly different sugar molecule.
So they're similar but not exactly the same.
Right. But the biggest difference is RNA's flexibility. It can fold itself into all sorts of shapes.
Like origami.
Exactly. This allows RNA to take on different roles in the cell.
So if DNA is the master plan, is RNA like the skilled worker who can do lots of different jobs?
Perfect analogy. You've got messenger RNA, or mRNA.
Messenger RNA, what does that do?
It carries the genetic code from DNA to the ribosomes.
Ribosomes. Those are the protein-making factories in the cell.
That's right. Then you have transfer RNA, or tRNA. It brings the building blocks of proteins, the amino acids, to the ribosomes.
And what about ribosomal RNA?
That's rRNA, and it's part of the ribosomes themselves. It helps assemble the amino acids into proteins.
Okay, I think I'm starting to get the picture here. We've got DNA, the blueprint, and RNA, the construction team, working together to build all the proteins that our cells need. But how does this system create all the incredible diversity of life that we see on Earth?
Now, that is a great question. And to answer it, we need to talk about the amazing variety of DNA sequences that are possible.
DNA sequences, what are those?
They're the specific order of those four bases, A, T, G, and C, along the DNA molecule.
So with just 4 bases, you can create all the different instructions for building every living thing.
It's kind of like a code, where different combinations of those 4 letters can create an almost infinite number of different words and sentences.
So different DNA sequences create different organisms.
Exactly. Think about it. Your unique DNA sequence is what makes you, you. No one else on Earth has the exact same combination of bases in their DNA.
Wow. That is mind-blowing. It's like a molecular fingerprint.
It is, and it's one of the most fascinating aspects of genetics. But I have a question for you now. If DNA is so unique to each individual, how is it that all living things seem to use the same basic system? from bacteria to butterflies to humans.
That's a great question. It's like we're all speaking the same language, even though we look and act so differently.
Exactly. And the answer lies in the genetic code itself. But to understand that, we need to dig a little deeper.
Okay, Mr. Martin's class, get ready for round 2 of our deep dive into the world of nucleic acids. We're about to crack the code of life itself.
Welcome back to the Deep Dive Mr. Martin's class.
Let's keep unraveling these mysteries of DNA. We've talked about its structure, how it replicates, but how does it actually store so much information?
Well, that's where the sequence of those bases, A, T, G, and C, come in. It's like a 4-letter alphabet that can spell out a huge variety of instructions.
Okay, so it's like a code where different combinations of letters make different words.
Exactly. And in DNA, those words are called genes.
Genes. Okay, so those are the segments of DNA that contain instructions for building something specific.
You got it. Genes hold the instructions for building proteins. And proteins are like the workhorses of the cell. They do all sorts of important jobs.
So one gene codes for one protein.
Well, it can be a little more complicated than that. Sometimes a single gene can actually code for multiple proteins, depending on how the instructions are read. But the basic idea is that genes are like the blueprints for all the different proteins that a cell needs to function.
Okay, I think I'm following. We've got these genes all lined up along the DNA molecule, but how does the cell know where one gene starts and another one ends?
That's a great question. There are specific sequences of bases that act like punctuation marks in the DNA code. They signal the start and stop of a gene.
So it's like periods and commas and things like that, helping us understand where sentences begin and end.
Exactly. Without those punctuation marks, the cell wouldn't be able to read the DNA code correctly.
So the DNA molecules have like a really, really long sentence with genes as the words and punctuation in between.
A very long sentence indeed. And the order of those genes and all the punctuation marks, it's crucial. It determines how the genetic information is read and used by the cell.
This is fascinating. Okay, so we've got the genes, the punctuation, but how does the cell actually use all this information to build the proteins?
That's where RNA comes back into play. Remember, RNA is like the messenger. It carries the genetic instructions from DNA out to the ribosomes where proteins are made.
Oh, right. RNA is like the construction worker who reads the blueprints and gathers all the materials to build the protein.
A very skilled construction worker. The process of making that RNA copy of a gene is called transcription.
Transcription, so it's like taking notes, copying down the instructions.
Exactly. It's like the cell is making a photocopy of a specific blueprint from that DNA library.
Okay, so the DNA stays safe and sound in the nucleus, and the RNA copy heads out to the ribosomes in the cytoplasm.
You got it. And then another process called translation happens once that RNA gets to the ribosome.
Translation, so now it's like the instructions are being deciphered.
Exactly. The genetic code is read, and the amino acids are assembled into proteins.
So transcription makes a copy of the blueprint, and then translation actually builds the protein based on those instructions.
Perfect. And all of this incredible process, is happening constantly inside every cell of your body.
Wow. I never really thought about it like that.
Yeah.
It really makes you appreciate how much is going on inside us all the time.
It really does. And the more we learn about DNA and RNA, the more we understand just how incredibly complex and beautiful life truly is.
You mentioned earlier that DNA can be used to store information. Is that like a sci-fi thing, or is this actually happening?
It's happening right now. Scientists are really exploring using DNA to store data. We're talking about storing massive amounts of information, libraries, maybe even movies, in a space smaller than a pinhead.
Wait, seriously? DNA can be like a hard drive.
It's incredible, isn't it? DNA is so dense and stable, we're talking thousands, maybe even millions of years of potential storage.
Whoa, that's mind-blowing. So we could store all of human knowledge in DNA, like for future generations.
That's the idea. It's still early days, but it's a really exciting field. Imagine accessing information from ancient civilizations or even extinct species by reading their DNA.
That would be incredible.
Yeah.
But it also makes you think about some pretty big what-if questions.
Absolutely. There are definitely some ethical considerations that come with such powerful technology.
Like, what if someone got access to information that they shouldn't? Or this technology could be used for something harmful?
Those are important questions to ask, but the potential benefits are also enormous, particularly in medicine, agriculture, and even environmental conservation.
It sounds like we're just scratching the surface of what's possible with DNA technology.
We are. We're living in a time of amazing scientific discovery, and who knows what breakthroughs lie ahead.
Okay, so this is all fascinating, but I have to ask, How does a sequence of just 4 bases create the incredible diversity of life we see on Earth? That's something I've always wondered about. Welcome back to the Deep Dive Mr. Martin's class. It's time to crack the code of life itself, the genetic code.
Yep.
I'm still amazed that just 4 bases can create all the diversity we see on Earth, from bacteria to butterflies to us.
It really is remarkable. Those four bases, A, T, G, and C, they're like letters in an alphabet. And they combine to form three-letter words called codons.
Codon. So each codon has like a specific meaning.
Yes, each codon corresponds to a specific amino acid, and amino acids are the building blocks of proteins. It's kind of like a recipe where each codon tells the cell which ingredient to add next.
Okay, so the DNA sequence is like this long recipe book filled with codons. And those codons tell the cell how to build different proteins.
Precisely. And here's the truly amazing part. This genetic code, the way these codons are interpreted, it's virtually universal.
Universal, you mean like across all life on Earth?
Exactly. Across all life on Earth, the same codons code for the same amino acids. So a codon in a human gene would have the same meaning in a bacterial gene.
Wow. That's incredible. So it's not just a code, it's like a history book telling the story of evolution.
It really is. And there's another interesting layer to this. With 4 bases and those 3-letter codons, you can create 64 possible combinations.
64. That's a lot of codons. But there are only 20 standard amino acids. So what happens to the extra ones?
Well, some amino acids are actually coded for by multiple codons. This redundancy is actually beneficial.
Beneficial. How so?
It helps protect against errors. During DNA replication or protein synthesis, sometimes mistakes can happen.
So even if there's a little mistake in the DNA sequence, it might not actually affect the protein that gets made.
Exactly. The genetic code is remarkably robust thanks to this built-in redundancy. It's like having backup systems in place to make sure the information is transmitted accurately.
It's amazing. It makes you appreciate the elegance and complexity of nature. So much thought went into designing this system.
It is truly remarkable. And the more we learn about the genetic code, the more we realize how much we still don't know. It's a vast and intricate system with many secrets still waiting to be unlocked.
Well, Mr. Martin's class, I think we've all had our minds stretched today. We've explored the structure of DNA, how it replicates, how it's transcribed into RNA, and the amazing complexity of the genetic code.
We've seen how DNA stores the blueprints of life, how RNA acts as the messenger and builder, and we even touched on the incredible possibilities of DNA technology.
From storing information to understanding our evolutionary history, DNA is truly a remarkable molecule.
And most importantly, we hope this deep dive has sparked your curiosity about the amazing world of molecular biology. Keep exploring, keep asking questions, and who knows, maybe one of you will make the next big discovery in genetics.
That's all for this deep dive, everyone. Thanks for joining us. We hope you learned something new and exciting about the amazing world of nucleic acids.